In this video, we are going to talk about the regulation of gene expression in eukaryotes. And really more specifically, we're going to be talking about multicellular organisms that are made up of eukaryotic cells. So remember that a eukaryotic cell is a cell that has a membrane-bound nucleus and membrane-bound organelles, such as mitochondria, endoplasmic reticulum, Golgi apparatus, etc., etc. Now, when we're talking about multicellular eukaryotes, for example, a human or a plant or some other animal, we're talking about an organism that is made up of multiple cells, oftentimes millions, billions, trillions of cells. And every single cell has a complete complement of the genetic material or the genome present. So remember, when we talk about things like chromosomes, we're talking about all of these cells having a complete set of the chromosomes. So remember that chromosomes contain the genetic material, so DNA. So DNA is packaged in chromosomes. And remember that an organism's genome is all of the genetic material that is being packaged or contained within those chromosomes. And for example, the human genome is made up of about 3 billion base pairs. And those base pairs, remember when we're talking about base pairs, we're talking about the DNA nucleotides. And the entire genome, so all of this information that is needed in order to make a person, is located in almost every single cell of the human body. There are some exceptions to that rule, including things like gametes, which only have half of the genetic information. But almost every cell has a complete genome kind of packaged in it, contained within these chromosomes. And remember that this genetic material, this DNA, is where we find our genes. And remember that genes are just these kind of recipes on how to make a protein through the processes of transcription and translation. But we know that different cells in the body have very different structures and functions to them. And that's because we might have let's say, a liver cell. And a liver cell is involved in digestion. It needs to be able to filter the nutrients that are being absorbed into the body. It also produces a lot of proteins that the body needs, especially in the blood. We could have a skin cell. Our skin cell has to have kind of structures that help it to protect the inside of the body. We might have a pancreas cell, and the pancreas is what produces the hormones insulin and glucagon, which helps to regulate blood sugar. We might have a muscle cell, and a muscle cell is involved in moving something around in the body. We could have a neuron. A neuron is involved in communication between different parts of the body. So we have all of these different cells, but they all have the exact same genetic information inside them. So why does a neuron look and function a lot different than a muscle cell? And why does a muscle cell look and function a lot different than a skin cell? And the answer comes in the form of the regulation of gene expression. So, different cells are going to regulate or turn on or turn off different genes depending on what is needed. So, a neuron is only going to turn on the genes it needs in order to function as a neuron. A pancreas cell is going to turn on different genes than a neuron because it has a different function. It doesn't need the same genes turned on that the neuron does, it needs different genes turned on. So, this regulation of gene expression really happens at the cellular or the tissue layer of organization so that individual cells can basically turn on the genes that they need in order to perform their function, but also turn off the genes that they don't need because that would be a waste of resources and energy making the proteins from genes that they don't need. Recall that during gene expression, a protein is being made. So we can kind of draw a shorthand of what is happening during protein synthesis by writing out this kind of simple equation of a molecule of DNA gets converted to a molecule of RNA, which gets made into a protein. And these arrows stand for the kind of different processes involved in this. So the first arrow stands for transcription, and the second arrow stands for translation. In eukaryotes, gene expression can be regulated almost anywhere along this process. So, for example, if we look at the DNA, so at the DNA, we're looking at genes. And remember that our DNA and these genes are kind of packaged in this substance called chromatin. And that chromatin can be condensed in order to form chromosomes. So the structure of chromatin can actually determine whether a gene is available to be expressed or not. Additionally, we have transcription. And remember that transcription is the process in which a molecule of RNA is made using DNA as a template. And so we can actually control the process at transcription. There's also a series of kind of alterations to the mRNA that happens before it gets kind of released to go out into the cytoplasm. So remember, there's also the post-transcriptional processing. And so we can actually regulate this process as to whether something gets transcribed in the first place and also what happens to it after it is transcribed. We can also control the process at translation. So remember that translation is where the mRNA goes out into the cytoplasm and it meets up with a ribosome and tRNA. And the mRNA is kind of converted, if you will, into a polypeptide. And then that polypeptide gets folded into the three-dimensional protein. So we can control the process at translation. We can also control it at the protein level. So we're actually going to talk about 5 kind of levels of gene expression in eukaryotes. And so we're going to talk about first the kind of the structure of chromatin and how that can determine whether a gene is expressed or not. We're going to talk about the regulation at transcription. We're going to talk about post-transcriptional processing and how that can be kind of changed or altered in order to determine whether a protein is made. We're going to talk about translational control. And then we're also going to talk about kind of a fifth layer here, and we're going to call this post-translational control. And this post-translational control kind of happens at the protein or the polypeptide level of production. So just to recap, the rest of this video is going to focus on the different levels of gene regulation in eukaryotes. So the first level is the structure of the chromatin. The second level is transcriptional control. The third level is post-transcriptional control. The fourth level is translational control. And the fifth level is post-translational control. The first level of eukaryotic gene regulation that we are going to talk about is the structure of the chromatin. And remember that chromatin is essentially DNA that is wrapped around proteins. And these proteins are called histones. How condensed the DNA is wrapped around these proteins can change depending on the needs of the cell. And so we actually have kind of two different types of chromatin, and they're really the same thing. They're still just DNA and proteins. The only difference is their structure and how tightly wound the DNA is around these proteins and how tightly wound the proteins are around themselves. So the two types of chromatin that we have, or the two basic types of chromatin we have, are euchromatin and heterochromatin. Euchromatin is more loosely arranged, which means that the genes in the DNA are available for transcription. And when we say available, we mean that they're accessible by the different proteins and enzymes involved in transcription. So the genes are available for transcription. Heterochromatin is more tightly packed. And in heterochromatin, the genes are not available for transcription. And it's important to note that euchromatin and heterochromatin exist typically on the same strand of DNA. So it could be that part of the chromatin is tightly packed and not available for gene regulation, whereas another area is more loosely arranged and those genes would be available for transcription. So it's not an all or none with regards to, say, the genetic information on a chromosome. It just might mean that part of the genetic information is being expressed and part of it is not, even if it's all on the same strand of DNA. There are actually a series of proteins called the chromatin remodeling complex. And these proteins are involved in basically loosening up the chromatin in the area where the genes need to be expressed. So these proteins actually open up or expose the gene. And once that gene is open or unpacked, it allows for transcription to occur. One really interesting kind of discovery in genetics that has really started to develop in the last couple decades is this concept known as epigenetic inheritance. And this is related to the structure of chromatin because epigenetic inheritance is variation in the inheritance patterns that is not due to differences in the DNA sequence. So previously, when we talked about Mendelian genetics, we talked about how we have two copies of every gene, and that one copy might be dominant and one might be recessive. And it's the interaction of the DNA sequence that determines what you look like and what your phenotype is. Epigenetic inheritance isn't necessarily a difference in the DNA sequence, but a difference in how the DNA is expressed. So, for example, if you were to take identical twins have the exact same sequence of DNA because they developed from the same egg and the same sperm. However, over time, especially, identical twins start to look and act differently from each other. And this is likely due to epigenetic inheritance, meaning that the genes that are being expressed in one twin might be different from the genes being expressed in the other twin. So, to recap, epigenetic inheritance is variation in inheritance patterns that is not due to the sequence of DNA. Or that is not due to differences in the sequence of DNA. Instead, we're looking at variation that is due to which genes are being turned on or turned off, meaning which genes are being expressed and which ones are not. And as I mentioned, this is a relatively new kind of field of study that's really kind of popped up in the last couple of decades. And so there's a lot of research going into this to try to better understand not only the genetic code, but how that genetic code is expressed. And so we don't know exactly how epigenetic inheritance is determined, but it may be inherited. So, along with your genetic information, how that genetic information is expressed might be also passed down from your parents, but it may also be influenced by environmental factors. And it is likely a combination of both of these. So it's likely something that has some inheritance value to it, but environmental factors can probably alter gene expression as well.